Traditional digital systems follow instructions linearly, executing fixed code with high speed and precision. By contrast, a biological neural network is adaptive, able to reorganize itself in real time, strengthen or weaken connections, and create entirely new pathways in response to stimuli. This fluid architecture is what gives rise to learning in organic brains, and in this new context, it offered a glimpse into how synthetic and biological intelligence might one day be combined. These experiments also raised important questions about the nature of intelligence and consciousness. If a dish of neurons can learn, remember, and act in ways that reflect intention, does that imply a kind of awareness? Or is it merely the outward appearance of learning, a reflection of mechanical responses refined through repetition? This boundary between organic behavior and conscious experience remains one of the central puzzles in neuroscience, philosophy, and cognitive science. The dish brain and its successor opened the door to exploring this boundary not through theory alone, but through living systems placed in experimental conditions. Another significant aspect of these projects is their potential for ethical inquiry. As neural networks become more sophisticated and capable of learning, questions arise about their treatment and rights. At what point does a network of living neurons deserve ethical consideration? Can pain, pleasure, or awareness exist in a disconnected biological substrate? While these questions do not yet have clear answers, the existence of platforms that bring them to the forefront of research means they must soon be addressed. From a technical perspective, these platforms also offer promising applications. Because they involve living cells, they can potentially serve as testing grounds for neurological treatments, the study of degenerative diseases, and drug testing that more closely replicates the behavior of the human brain. They may also serve as models for developing new forms of adaptive computing that do not rely on silicon-based architecture. In this sense, the project bridges several disciplines, from bioengineering and neurology to artificial intelligence and ethics. Looking ahead, the continued exploration of such systems may result in entirely new classes of machines that are neither fully organic nor entirely digital. These hybrids could reshape computing, creating systems that are not just tools, but adaptive partners in solving complex problems. Whether these systems remain confined to laboratories or one day find their place in broader society, they represent a fundamental shift in how intelligence is conceived, built, and studied. Recent developments in synthetic biology and sensory technologies have brought together several once separate disciplines, leading to innovations that are beginning to reshape how machines perceive, adapt, and respond to the world around them. At the heart of these developments are living neural networks grown in controlled environments and connected to responsive platforms. These networks, as seen in experimental projects involving cultured neurons in laboratory dishes, have demonstrated the ability to learn, adapt, and respond to external stimuli in real time. By linking the biological flexibility of living brain cells with a digital framework capable of interpreting and reinforcing behavior, these systems have revealed unexpected levels of intelligence in organic substrates outside of the body. The work has continued with advanced platforms that expand the complexity of the environment, enabling the neurons to process greater volumes of information, retain experiences, and develop behavioral patterns that hint at rudimentary awareness. In parallel with this, another class of technology has begun to focus on sensing the invisible chemical fingerprints that the human body and other organic materials release. These technologies are based on the principle that biological decay produces specific chemical markers, often in the form of airborne plumes, that can be detected at a distance. When the body breaks down, it emits a sequence of volatile compounds, many of which can be traced back to precise processes occurring in soft tissue, blood, or microbial activity. By identifying the patterns in these emissions, systems can detect the presence of a deceased individual even if the location is hidden, submerged, or buried. The connection between the two fields becomes especially powerful when the adaptable learning capacity of biological neural networks is brought into contact with chemical plume detection. Traditional chemical sensors rely on fixed parameters, reacting only to pre-identified compounds. But systems informed by living neural networks can begin to identify more complex and subtle signatures, even when traditional machines struggle to recognize them. A network trained through exposure to varied biological decomposition scenarios can learn to distinguish between natural decay, environmental interference, and the specific profile of human remains. These synthetic biological platforms are capable of adapting to uncertainty in a way that conventional systems cannot. They do not require rigid code, instead, they learn from feedback, building memory through lived experience. A system trained in this way can not only detect the presence of volatile organic compounds associated with human decomposition but can also differentiate between the decay of a living organism and other natural processes, such as plant rot or industrial chemical leaks. This is especially useful in forensic and search and rescue scenarios, where time, terrain, and ambiguity challenge the limitations of standard detection equipment. The sensors used for these purposes typically monitor the air for a mixture of compounds such as ammonia, cadaverine, putrescine, sulfur-based gases, and other volatile organic substances that are consistently released during the breakdown of human tissue. The unique blend and ratio of these gases create a signature that, once learned, can be recognized by the integrated system. When this data is processed by a biological learning model like a cultured neural network, the system becomes capable of not just recognizing known patterns but of identifying new, previously unencountered chemical profiles with a degree of inference and context.